Bismillahirrahmanirrahim, and welcome back to this next video. And this is yet another video in the series of videos on the digestive system of the human beings. In this particular video, I want to focus on the functions of the gallbladder and how the gallbladder is helping in the process of the digestion. So the gallbladder, that is also known as the cholecyst, it is actually a very small pouch that sits just under the liver. As you can see over here, this is the liver and this is the position of the gallbladder. So it is sitting just under the liver. Now what the gallbladder do is that it stores and it concentrates the biles. Actually the bile is produced by the liver, but when it is produced by the liver, it is transported to the gallbladder, and the gallbladder actually acts as a storage house for the bile, and it is also responsible for the concentration of the bile before it is released into the small intestine. So this is the function of the gallbladder, that it's going to store and it is going to concentrate the bile, which is actually produced by the liver, and when it gets concentrated, it is released into the small intestine to help in the process of the digestion. Now after meals, the gallbladder is empty and flat like a deflated balloon, and the reason is very obvious, because the gallbladder have released all of its bile content into the small intestine, so its shape is like a deflated and it is like a flat in structure. Now before the meal, as the gallbladder have bile in there, so the gallbladder may be full of bile and about the size of a small pear. So this is the location of the gallbladder in the body. Now what the gallbladder do is that it is going to receive and store the bile from the liver via the common hepatic duct. So when the liver that produces the bile, that is transferred into the gallbladder, what is via the common hepatic duct, as you can see over here. So this is actually the duct leading from the liver into the gallbladder, and the bile moves through this common hepatic duct from the liver into the gallbladder. And when that is stored and when that is concentrated, the gallbladder is going to release it into the duodenum via the common bile duct. So this one is the common bile duct. So this duct is actually releasing the concentrated bile into the duodenum, and the common hepatic duct is actually going to release the bile from the liver into the gallbladder. Now when you talk about the composition of the hepatic bile, it contains a lot of things. Mostly it's water, like 97 to 98% is water. There are bile salt, the total composition is like 0.7%. There is bilirubin, which is actually a coloring pigment, and is actually also a breakdown product of the red blood cells. There are fats in the composition of the hepatic bile, the phospholipids, and there are also small quantity of the inorganic salts. Now when you talk about the healthy individuals, about 400 to 800 milliliter of bile that is produced every day in the adult human beings. Now let us focus on the function of the bile, how this bile works. Now this bile is actually responsible for the digestion of the fat that you take in your food, and the most common fat molecule that is present in the food, they are the triglycerides. Now these triglycerides, they are actually hydrophobic in nature, because if you see the structure of the triglyceride, there is a glycerol molecule, as you can see that over here, and it is attached to three free fatty acid molecules. So this is one free fatty acid molecule and this is a glycerol. So when they combine with each other, one glycerol in three free fatty acid, the structure you are going to get, that is known as the triglyceride, and it is the most common fat molecules that we get through our food. Now what happens is that there is an enzyme which is known as the lingual lipase, and this lingual lipase that is produced by the salivary gland, but it is activated in the stomach, because for the activation of the lingual lipase you need an acidic pH. The pH is not acidic in the oral cavity or in the mouth, therefore the lingual lipase is produced by the salivary gland, but it gets activated in the stomach. So when you take the triglyceride in the food, the lingual lipase and that triglyceride is going to move into the stomach. in the stomach, the lingual lipase is going to get activated because of the acidic pH of the stomach, and the stomach is also producing a lipase which is known as the gastric lipase, and the gastric lipase is actually produced by the chief cells of the stomach. Now, what are the chief cells? They are actually the enzyme producing cells of the glands that are present in the stomach. I have a detailed video on those glands and I'll share the links in the description. But simply, if you want to differentiate between the two, this is the lipase, but the term we are using the term lingual over here. It simply means that it is produced by the salivary gland, and when we use the term gastric with the lipase, this means that this is the lipase which is produced by the chief cells of the stomach. So the lingual lipase and the gastric lipases, they are going to start the digestion of the triglyceride by breaking some of them into a diglyceride and free fatty acid molecule, and about 30% of the triglyceride that is broken down into diglyceride and fatty acid in the stomach. What I mean by the diglyceride is that you will be having a glycerol molecule and 2 free fatty acid that will be attached to them. So this one free fatty acid that has been removed from it, so this will then become a diglyceride. So is the di mean 2, so 2 of the free fatty acid molecule that will be attached to the glycerol molecule. So 30% of the triglyceride that will be broken down by the lingual lipases and gastric lipases in the stomach. Now the triglycerides, they actually then enter the small intestine as large lipid biomasses because of their hydrophobic nature. The environment in the small intestine is hydrophilic. These triglycerides, they are hydrophobic in nature. So what they do is that they are going to make these large fat globules, and they are going to make this large massy structure because of their hydrophobic nature. Now what happens in the small intestine is that the bile is going to that the gallbladder is going to release the bile, and that bile is actually responsible for the breakdown of these large fat globules into small molecules or the small fat droplets, which are known as the emulsion droplets. And this particular phenomena in which the bile is going to break down the fat globules into these emulsion droplets is known as the emulsification. So in the process of the emulsification, the bile salts and the phospholipids, which are actually the component of the bile, they are going to interact with these large fat globules, and they are going to convert them into the what are known as the emulsified fat droplets. As you can see over here, if this is a very large structure, a large fat globule over here, so the process of emulsification in which the bile is interaction with these large fat molecule, they are converted into the small emulsified fat droplets, and they are known as the emulsion droplets. So in the intestine, the small intestine, the first step is the process of emulsification, in which you are going to break down these large lipid biomasses into small molecules, which are known as the emulsion droplets. Now the pancreas, that is also going to release a lipase, and that particular lipase is known as the pancreatic lipase. So as you can see, a lot of lipases are involved. So to differentiate between these lipases, the one produced in the produced by the salivary gland is known as the lingual lipase. The one produced by the stomach is known as the gastric lipase, and the one produced by the pancreas is known as the pancreatic lipase. Now what the pancreatic lipase do is that they are very active in interacting with these small molecules, these small emulsion droplets, and when they act on them, they are going to convert them into the monoglycerides and the free fatty acid. And the monoglyceride simply mean glycerol molecule attached to a single fat molecules, and the other fat molecule have been released from the diglyceride. So the pancreatic lipase that act on these emulsion droplets, producing the monoglyceride and the free fatty acid. And in this particular phenomena, the bile salt and the phospholipids, they are going to interact with these monoglycerides and the free fatty acids, thereby making a structure which is known as the micelles, and these micelles are much smaller than the emulsion droplets. So as you can see, you are going from a very large structure into somewhat a smaller structure, and then these smaller structure are further converted into much smaller you can say components, which are known as the micelles. So you are going from the large lipid biomasses to to the emulsion droplets, and the emulsion droplets are converted into much smaller components which are known as the micelles. Now what the micelles do is that these micelles, they transfer the monogallbladder and the free fatty acid which are produced by the pancreatic lipase to the membrane of the enterocytes in the small intestine. And at the membrane of the enterocytes, they enter into the enterocytes, and when they are there in the enterocytes, they are going to form the triglyceride again. Now the triglyceride, when they are made inside the enterocytes, which are actually the cells of the small intestine, so the triglyceride, they are going to combine with the proteins, thereby making a structure which is known as the lipoprotein. Sometimes it is also known as the chylomicron. So the first thing you need to understand is the difference between the micelles and the chylomicrons. So the key difference between the micelle and the chylomicron is that the micelle are globule of lipid molecules that are arranged in a spherical form in an aqueous solution. As you can see over here, the micelle have got a hydrophobic head which will be towards the aqueous solution, and they have got these hydrophobic fat molecules which are inside this particular structure. So they are going to make this spherical structure in the aqueous solution, with the hydrophilic head being exposed to the aqueous solution, while the hydrophobic part that is present inside this particular structure. And when you talk about the chylomicrons, they are actually the lipoproteins that consist of a core made from the triglyceride and the cholesterol. You can see over here, the C is for the cholesterol, this T is for the triglyceride. So the core is actually made up of the triglyceride and the cholesterol, but their coat is made up of different kind of the proteins, like you have got like apolipoproteins like ApoB, the ApoA, the ApoC, the ApoE. So the coating is actually made up by these particular apolipoprotein and phospholipids, but the core that is made up of the triglyceride and the cholesterol. So this is the major difference between the micelles and the chylomicron. Now, when these chylomicrons, they are made inside the enterocytes, then they are transferred into the bloodstream via the lymphatic vessels, and when they are there when they enter the bloodstream via the lymphatic vessels, they are transported to liver, to fatty tissues, to muscle tissues and other tissues in the body, where they are utilized for the normal physiological functions of the body. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.